In this video, we're going to review DNA replication. Think back to the cell cycle. DNA is necessary for cells to function, and if we need more cells, then that means we also need more DNA. So DNA replication happens before a cell can divide, which means in the cell cycle, it has to happen during interphase, and more specifically, during the S phase of interphase. S, remember, stands for synthesis. So DNA synthesis we also call DNA replication, which means to copy. Where do you think DNA replication happens? Well, where is DNA kept? Inside of the nucleus of eukaryotes. So DNA replication in eukaryotes always happens inside of the nucleus. Now let's review the 4 steps of DNA replication. DNA is all jumbled up and mashed into the nucleus of the cell. So it's twisted, and we want to get to the important information that's inside of the DNA double helix. So we first unwind or untwist. Then the genetic information is stored on all of the nitrogen bases, so we want to unzip. This is going to happen using an enzyme called helicase. Helicase then goes in and acts kind of like a hungry hippo, chomping through all of those hydrogen bonds to unzip or separate the 2 sides of the DNA. So once we have our exposed bases, we have the free-floating nucleotides that match on with their complementary base. A matches with a T, C matches with a G, and so on and so forth until we have both of those exposed strands completely filled up with the nucleotides that match with them. Then, an enzyme called ligase helps to make those hydrogen bonds between them and kind of zips them back up, so we're left with 2 identical strands of DNA. This process is called semiconservative because half of each of these identical strands is old from the parent DNA strand, and half is new. So it's semiconservative because it keeps half and then makes half of it new. Let's go through this and review with another one of our little dances. DNA first unwinds, then it unzips, and our free-floating nucleotides bond on using ligase to form those bonds again, and we're left with 2 identical DNA strands. All right, so let's go ahead and look at an example of this. This is a kind of problem that you might see in the future on STAR tests or things like that. They'll give you the template strand. This is going to be already when the DNA has unwound and unzipped, and it's giving you 1 strand of DNA, and you're supposed to go through and put the matching nucleotide with it. So let's review the base pair rule again real quick. A pairs with T, and C pairs with G. All right, so using the base pair rule, we're going to go ahead and make our complementary strand. So A must pair with a free-floating nucleotide, T. Another T. And then here, along with our T on our parent strand, we're going to add A, adenine. The C is going to be paired with guanine. The G is also paired with cytosine. A, T, G is paired with C, and then our last T is paired with a free-floating A. As long as you know the base pairing rule, it's easy peasy. You can do them back and forward in your sleep. Please don't forget to look at our resources in the folder. This wraps up our video of replication.